Okay, let's talk about some physiology that occurs at capillaries. First, we see in this drawing that blood's going to enter the capillary from an arteriole, right? So this is one of those small little arteries. The blood enters the capillary. And this first part of the capillaries are known as the arterial ends, all right, because the blood enters from an arteriole. Then we have what's called the venous end or venous capillary right here because it's going to then lead to a venule and head back in a vein. We could call this kind of a capillary network or a capillary bed. So the blood goes through all these little capillaries, but we also want to be able to control how much blood. So there's a couple ways to change how much blood goes through these capillaries. One is you can dilate the arterioles if you need more blood. So if you get these arterioles bigger, more blood can enter the capillaries. There are also what's called precapillary sphincters. So these are these little rings of muscle that can close off capillaries or open them up. So if this was, say, capillaries of a skeletal muscle, let's say these capillaries are in muscles of your legs, and you're walking or even exercising, we're going to want to send more blood through that tissue. We call that perfusion. Perfusion is, sending blood into a tissue. So we want to increase perfusion of that skeletal muscle. So this arteriole is going to dilate. These precapillary sphincters are going to relax, and we can send more blood through these capillaries. Then, once we're not exercising and say we're resting, that muscle doesn't need all that perfusion. So this arteriole can constrict and get smaller, and then we can actually close off some of these capillaries and send less blood through that capillary bed. All right, so that's one way we can regulate how much blood flow goes to an organ. Let's now talk about exchange. So how do things get exchanged? You'll notice these little arrows show that, okay, at these arterial end, we're going to want to deliver stuff, right? So this blood's going to have oxygen and nutrients. So we want to get that stuff out of the blood so it can go into the tissue cells. And then at this end, we start to bring stuff back in. Right? We're going to bring in carbon dioxide, bring in any kind of waste. All right? So we deliver stuff at the arterial end, we pick up stuff at the venous end, and then we head back. So how do we do that? Well, one way is by diffusion. So if you remember from maybe a basic biology class or from A&P 1, diffusion is when there's molecules that we have a high amount of them in one location, and then they're going to move towards an area of low concentration. So that's called moving the substance down a concentration gradient. So if you think of oxygen, right, there's going to be a lot of oxygen in this blood, but if there's cells here, those cells have less oxygen because they've been using it for cellular respiration, ATP production. Same thing is true with glucose. You're going to have more glucose in the blood than you do in the cells because they've been using glucose for energy. So molecules like oxygen, glucose, amino acids, hormones can all go by diffusion. They're going to go from the blood into the tissue cells. Carbon dioxide is going to go the other way because cells are going to have high amounts of carbon dioxide because that's one of their waste products of cellular respiration. The blood's going to be lower in carbon dioxide, so carbon dioxide is going to be a molecule that diffuses from the cells into the bloodstream. So diffusion is an important mechanism. We also use what's called transcytosis. This is basically moving things in and out of cells using endocytosis and exocytosis. So the idea here is you can bring something through a cell by bringing it into the cell and then out of that cell. So think of the wall of a capillary, right? The wall of a capillary is a single cell. It's an endothelial cell. So that cell can bring something in and then out in order to cross that cell and get either into the blood or out of the blood. So that's known as transcytosis. This is how we might move large molecules. This is also how we move other things like such as insulin. All right, and then the major way how we move a lot of stuff is called bulk flow. And this is one of these physiological concepts which is going to be a little tricky, but it's really important for understanding some of the clinical things that occur, such as edema. So this is going to help us understand why people develop edema. So bulk flow, the driving force is pressure. So if we have pressure, we can push fluid out of the blood, and in that fluid is going to it's going to contain a lot of molecules that you're trying to deliver. So if you can have a higher pressure on the inside of the blood than outside, you can actually push fluid out of the blood. And then at this end, the venous end, if we can change that pressure, all of a sudden we have a lower pressure in the blood versus outside. We can push stuff into the blood. So that's called bulk flow. That's what this figure is going to help us figure out. All right, so let's kind of walk through this figure. This is a capillary. This is how blood's kind of entering from an arterial. So this is the arterial end. All right, then this is the venous end because then blood enters a venule. There's a couple important forces or pressures that I want you to understand. The first one is called hydrostatic pressure. Hydrostatic pressure is blood pressure, all right? Now, it's not the 120/80 because we're not in an artery, all right? It's much lower. In this case, we're going to use 33 millimeters mercury. And if you remember, if you refer back to that blood pressure graph, that was the pressure at the start of a capillary, all right? Blood hydrostatic pressure, all right, is a pushing out of the blood force. All right, so blood hydrostatic pressure pushes fluid out of the capillary. Okay, now there happens to be an opposing force, all right, called osmotic pressure. All right, osmotic pressure is due to the protein inside the bloodstream. All right, so because you have a lot of protein in your blood, it's going to pull water into the bloodstream. It kind of acts like salt, right? If you have high salt somewhere, water likes to travel towards that salt. Same thing is true here with the osmotic pressure. It pulls fluid into that capillary. All right. So we have hydrostatic pressure, also called blood hydrostatic pressure, or BHP. And then we have osmotic pressure. Another name for that is colloid osmotic pressure. They're opposing forces. So one's pushing out, one's pulling in. But at this arterial end, notice who is bigger. It's the hydrostatic pressure. So if you determine the net, so net is always when you kind of add things together, but in this case, since they're opposite, you kind of have to subtract, right? Because they're not additive, they're acting against one another. So if you have a force of 33 pushing out, but you have a force of 20 pulling in, your net filtration pressure is 13. So what that means is you're actually going to push fluid out at this arterial end, okay? Once you keep going, remember what happens to blood pressure. Blood pressure drops as you go away from the heart. So if you get to this venous end, the osmotic pressure hasn't changed. It's still 20, all right? But the hydrostatic pressure has dropped to 13. So now you have a higher osmotic pressure compared to the hydrostatic pressure. So the result will be more pulling in, or a negative net filtration pressure. All right? So you push out fluid, and then you pull in fluid. And we have names for that. When you push fluid out of a capillary, it's called filtration. When you pull fluid back into a capillary, it's called reabsorption. So right now you should be writing those two words down because I don't have them on the slide. So at the arterial end, you filter. So filtration is pushing fluid out. At the venous end, you reabsorb. You pull fluid in. Now, it's not the same fluid, right? The fluid that gets pushed out has nutrients, things you want to deliver. The fluid you pull back in has the waste, all right? Now, there's a problem. This is not balanced, right? This is not homeostatic. There's no homeostasis here because you're actually pushing out at a 13 millimeters mercury. You're pulling in at a 7. There's an imbalance. So about 1/10 of the volume that gets pushed out here has to be collected by lymphatic capillaries, or you would have too much fluid accumulating outside of the capillary. All right, so this tells us the reason why we have to have lymphatic capillaries. Lymphatic capillaries readjust this imbalance in fluids. All right, if you just let this be, you'd be pushing out more fluid than you can pull back in, and you would develop what's called edema. Edema is an accumulation of interstitial fluid. But in order to prevent edema, we have the lymphatic capillaries collect that little extra fluid, drain it out of that tissue, and eventually bring it back into the bloodstream. And we'll talk more about lymphatics in the next chapter. So it's a pretty difficult concept. There's some animations that I'm going to have you watch that help this concept. But the idea is we're using pressure, all right? We're using hydrostatic pressure and osmotic pressure to better understand why we can force fluid out at the arterial end and then pull fluid back in at the venous end. One other thing we better make sure we understand is that the reabsorption process, this osmotic pressure, is largely due to the protein in your bloodstream. Knowing this, we can understand edema. So edema is an imbalance. So it could be either an excessive amount of filtration. All right, so now if you filter even more, you'll push even more fluid out. And that fluid accumulates in the interstitial fluids. This can be due to hypertension. So people with hypertension are more prone to developing edema because they have a higher hydrostatic pressure. So if I just click back real quick, if this number was even higher, right, because someone has hypertension, then this number is going to be even higher than this number. So you're going to get even more filtration and you just can't reabsorb enough. So you develop edema. Another cause is instead of excess filtration, you might not have enough reabsorption. And that can be due to losing protein. Remember the idea here. We got to make this connection. Protein, plasma protein, such as that albumin, allows for that osmotic pressure. It pulls fluid back in. But if you don't have enough protein, then you can't pull that fluid back in and you can develop edema. Protein, if you remember, comes primarily from your liver. So if someone has liver disease, hepatitis, cirrhosis of the liver, they're not making enough albumin from their liver, they don't have enough protein to reabsorb, they can develop edema. Burn victims, this happens to burn victims as well. They develop a lot of edema due to the loss of protein in the blood. Malnutrition, so let's say you don't have protein in your diet. If you're not, consuming protein, you're not going to have enough amino acids to build your own protein. And then finally, sometimes kidney disease can cause this because the kidneys, if they're sick, if you will, they'll actually urinate out protein. And you'll lose your plasma proteins because your kidneys. All right. So this all centers around this concept that the plasma proteins help pull the fluid back into the bloodstream, which is called reabsorption. So if you don't have those proteins, you can develop edema. And then a third cause of edema is defective lymphatics. So remember, the lymphatics are draining that extra fluid and putting it back into the blood. Well, if you have some sort of blockage you can't drain that fluid out of the tissue and you can develop edema. And that's what is shown here in this arm. So this person must have had some blockage or removal of the lymphatics. What it probably was from is removal of lymph nodes and lymphatics because of cancer. A lot of times if someone develops breast cancer, you got to remove a bunch of lymph nodes because the cancer will be found in the lymph nodes. And if you remove those lymph nodes, you just disrupted the drainage from the arm on the same side. So this woman must have had lymph nodes removed from her armpit on this left arm, and now the fluid is not draining out of that arm properly, and she's developing what's called lymphedema. So it's a pretty complicated process, this capillary exchange, but if you get to the point where you understand it, then you'll understand more important clinical connections when it comes to understanding this accumulation of interstitial fluid, which is called edema. All right, next topic before we kind of get into control of blood pressure is bringing blood back. So we just talked about capillaries. Well, after the capillaries do their thing, we got to get that blood back to the heart, and that's known as venous return. Now, the problem is the pressure's so low, right? By the time we get to veins, the pressure is very low. So how are we going to get that blood flowing up to the heart when we have such low pressure? So a lot of times we need help. So this kind of explains what helps promote venous return. There's three things. One is you got to have a nice pressure gradient. So if you've got a nice pressure gradient, blood flows pretty well. So the gradient we're talking about is actually the 13 at the venules and the 0 at the right atrium. That's a pretty good gradient. So blood should flow from this high 13 to this low 0, which is nice. Now, what might happen? Well, this number could get low, or this number could get high. And if this number gets low or this number gets high, the gradient gets smaller, and it's harder to bring blood up to that heart. One cause of this number going up is a leaky tricuspid valve. So if someone has a tricuspid valve on the right side of the heart that's leaky, that blood trickles up to the right atrium and causes the pressure to go up. And now it's harder to get blood to enter that right atrium. So that's an example of a disruption in the pressure gradient causing the blood to have a hard time getting up and into that right atrium. One other thing we have that helps is skeletal muscle. It's called the skeletal muscle pump. So that's what this picture is showing. So remember, veins have valves. And if you could squeeze on those veins using muscles, that blood will get pushed. And as long as those valves are working, that blood should get pushed up, but won't get pushed down because of that valve closing. So this is why it's recommended that people with poor circulation, low blood pressure, elderly, walk around more. Because if they don't walk around, you don't have these muscle contractions squeezing on those veins. All right? So that's called the skeletal muscle pump. There's also what's known as the respiratory pump. A similar process happens in the veins of your chest. But instead of muscles, it's when you breathe. So when you breathe, the pressure in your chest increases and decreases, which will squeeze on the veins. So squeezing veins is a good way to help that blood get pushed up towards the heart. All right, so that's called venous return. All right, we'll end that video. The next video, we're going to talk about how the nervous system controls your blood pressure, and then we'll move into how hormones control your blood pressure.